start? Excellent. You can share your screen whenever you're ready. Okay, so it is my very great pleasure to introduce our final speaker in the Diversity in Biology series. It's been a really excellent series so far. Before I do, I'm just going to say my name is Robin Leung, and I'm one of the organizers of the committee. We always start with a land acknowledgement. So I want to start that by acknowledging the territories, the ancestral territories of those whose land we're currently living and working on. A lot of people that attend this seminar are from UBC Okanagan, so and in Kelowna. So this is the ancestral and unceded territory of the Syilx Okanagan Nation. Of course, Dr. Leung and many other people who join us may not be in Syilx Okanagan Nation territory. So we always encourage you to reflect on the lands in which you live and work and whose the ancestral caretakers of those lands might be. I also want to acknowledge the funding sources for this. The series is funded by the UBC Anti-Racism Initiatives Fund, as well as the UBC Okanagan Department of Biology and the UBC Okanagan Irving K. Barber Faculty of Science. And so with that, I will introduce our speaker, who is Dr. Janice Leung. And so Dr. Leung is a clinician scientist at the University of British Columbia, Vancouver campus, where she both has a clinical practice devoted to respiratory care of patients living with HIV at St. Paul's Hospital, and also a translational laboratory at the Center for Heart and Lung Innovation. She is a Tier 2 Canadian Research Chair in Translational Airway Biology, and her work is supported by the Canadian Institute of Health Research, the Michael Smith Foundation, Genome BC, and the BC Lung Association. I think I got all of those in there. So that's amazing. And her talk, of course, as you can see on the screen, is titled Insights into Chronic Lung Disease in People Living with HIV. And so I pass it over to you, Dr. Leung.
Thank you so much, Robin. And I would just want to thank you and the organizers for inviting me to the seminar series today. I was just saying that it's a great idea, and I feel very privileged to take part today. I know you are all in the Okanagan. I am in Vancouver. And I just want to acknowledge myself that I am on the traditional unceded territories of the Musqueam, the Squamish, and the Tsleil-Waututh nations. These are my disclosures, some of the grant funding that I've received. And my objectives today, I'm going to first start talking about how aging with HIV has impacted respiratory health, and how my experience as a clinician has then informed some of my research. And in particular, the next part of my talk will be discussing some of the mechanisms of lung injury and HIV. And I want to focus on my lab's interest in accelerated aging and microbial dysbiosis in this population. And I'll highlight some of the work of my graduate students along the way. All of my work takes place at St. Paul's Hospital. This is a tertiary care center situated in downtown Vancouver. I have my clinic here, and I also have my lab here. And for someone who is interested in HIV-related lung disease, this really has been a wonderful place to work because it is at the intersection of many different disciplines. So I have my clinic at the Pacific Lung Health Center. I have a clinic that is both general respirology, but also specifically focused on the patients living with HIV who may have chronic lung diseases. And I get most of those referrals are sent to me. So I get to interact with these patients, and my interactions with these patients then inform my research. So my research lab is also at St. Paul's Hospital at the Center for Heart Lung Innovation. And I like that I'm situated so close to my clinic. I'm situated close to our bronchoscopy suite, which I'll discuss, which has become an essential part of my platform. I get access to patients, access to patient samples. And so it's been really nice to have that close proximity. And the other aspect of St. Paul's that's been really great is it's also the site of the British Columbia Center for Excellence in HIV/AIDS. And this has been, a world-renowned HIV research center. They take care of patients at the John Rudy Immunodeficiency Clinic. I have had wonderful collaborations with researchers and clinicians there. And so it's really just a place of synergy for someone who has interest in both HIV and respirology. One of the things that I've noticed taking care of patients with HIV over the years is there is this common theme. I hear this a lot. And it's patients who tell me, I never expected to live this long. And how this statement has impacted their care and kind of the transitions as we have gone through the AIDS epidemic has been very, very interesting from a specialist perspective. I dug back into the historical record, and if you go all the way back to 1981, this was the first sign that something was up, right? This was the report by the Centers for Disease Control about this outbreak of pneumocystis pneumonia in Los Angeles. And pneumocystis, is a fungal infection that only really affects people with compromised immune systems. And because it was happening in these 5 young, otherwise healthy men, it was very concerning. And this was the first clue that something new might be arising. Of course, by 1983, research scientists had discovered the human immunodeficiency virus, which was the cause of AIDS. And at that time, the reason why patients are telling me I never thought I would live this long is for many years, this was the face of AIDS. This is a very powerful photograph that was published in Life magazine in 1990. This is David Kirby. He is a young man. He was a young man. He was dying of AIDS. This is a picture of him on his deathbed, surrounded by loved ones. And it really brought home the message that, this is a real epidemic for real people with real consequences. And given all of the stigma and all of the discrimination of that era, which to be honest, is a lot of that still exists today, it really brought home a real message that this epidemic had a human cost. But for many patients, this was their fate, right? If you were diagnosed with HIV/AIDS in the 1980s, this is what you had to look forward to. And there was no cure at that time. There remains no cure, but with scientific progress, things have changed drastically. So by 1987, you had the first anti-HIV drug. This was a repurposed cancer drug called AZT. It didn't work that well. There were a lot of side effects, but it showed promise against HIV. And by the mid-90s, scientists had discovered that if you could combine different classes of HIV therapy, you could start to control the disease. And so, just for some a local historical interest, that announcement was made in Vancouver at the International AIDS meeting here in 1996. And of course, this was a game changer, all right? So if you look at British Columbia over the last 2 decades, there has been an amazing drop in HIV-related mortality, going from kind of the peak in around the early 2000s, getting people on combination antiretroviral therapy, and you can see the numbers have just fallen. I was a resident around this time period here, and I remember that we would have a rotation on the HIV service, and actually by this time it was we'd consider it a very easy rotation, because there were fewer admissions, you could get a good night's sleep, and it's really just a wonderful testament to scientific progress how the care of HIV patients has changed over these decades. And what this has meant is that there's been a shift in the demographics, right? So if you look at the more elderly populations, over 65 here in red, 45 to 64 here in the purple, there's been an expansion of that demographic group as these patients have started to age. Now, lifespan is not the same as health span though. So while the top 2 lines are the years expected that you would live if you were diagnosed with HIV at the age of 21. And, with HIV here and without HIV, the gap is starting to close. It's not quite there yet, but it is starting to close. But if you're looking at comorbidity-free years, you can see that there is still a gap here. And so aging with HIV comes with its own complications. There is a lot of multimorbidity, so conditions like diabetes, hypertension, chronic lung disease, my interest, stroke, cancer, frailty, coronary artery disease, chronic kidney disease, cognitive impairment, and dementia. All of these are things that we now have to consider in the chronic care of patients living with HIV. So as an example, I'll just show you a CT scan from one of my patients who is 45. He has well-controlled HIV on, takes his combination antiretroviral therapy without any interruptions, good CD4 count, undetectable viral load, and not a smoker. And let me just show you his CT scan. Now, you don't have to be an expert radiologist to know that this is not a normal CT scan. If these are the lungs, you see these basically holes. There are holes in his lungs at the age of 45. This is something that I would see in maybe an advanced smoker who might have been, 70s, 80s. But I will tell you that this patient is not uncommon in my practice. And, the fact that it's happening at the age of 45 is pretty concerning. He has chronic cough, chronic shortness of breath. And so, this is very, very unusual. And so what he has is pretty early onset chronic obstructive pulmonary disease, or what we call COPD. Now, this is a progressive disease of persistent airflow obstruction. Your airways fill with mucus. You get these expanded air pockets called emphysema, where all of the alveolar attachments are disrupted, inflammation, periodic events where these patients exacerbate and require steroids and prednisone. So it's a very debilitating disease. And unfortunately, I say this as someone who does this for a living, it's very hard to treat. We do not have the kind of treatments that other diseases may have. A lot of our treatments just sort of treat the symptoms, but don't really change lifespan, mortality. It's still, there's still a space for therapeutic discovery here. And so it's quite disturbing to see this happening to him at the age of 45. In general, if we talk about Canadians on a whole, COPD is very common. It affects at least 2.6 million Canadians now, and it accounts for 11,000 deaths and 90,000 hospital admissions per year. And it is actually the leading cause of hospitalizations if you discount childbirth in Canada. So there's a big health and economic impact for COPD. Now, in people living with HIV, though, these are data from my colleague at the BCCFD, Dr. Viviani Lima. If you look at the age and sex standardized prevalence compared to the HIV negative population, it's higher in patients living with HIV. And as well, she's found that the onset is at least 6 years earlier than in HIV uninfected individuals. So it's affecting more and it's affecting earlier. What is interesting about this population, unfortunately, is that they are very symptomatic as well. So this is a study that I did back when I was a postdoc fellow here, and we interviewed patients living with HIV in Vancouver, and we did something called the St. George's Respiratory Questionnaire Score, which really asks questions about how breathing and respiratory symptoms impact their lives. And the higher the score on this scale, the worse your respiratory symptoms are and the more impact that they have on the quality of your life. And I plotted this against lung function here. So this is a test that we can do to see how well the patient can exhale, and it's a measure of obstruction. So the higher your number, the better the lung function. A normal St. George's score would be less than 10, and I've also marked what I would consider to be a normal lung function here. And you can see that in our HIV population, a lot of people have very abnormal scores. And what's more, they have abnormal scores, even though their lung function might not be so bad. So it's a very symptomatic population from a respiratory standpoint. And so these patients are dealing with things like cough on a daily basis, wheezing, sputum production, and diminished exercise capacity. So the question that my lab has been asking is, why are they more susceptible to early and more severe manifestations of chronic obstructive pulmonary disease? We have to kind of go back. A lot of people ask about smoking, right? So is this just a reflection of smoking habits in HIV? And yes, the prevalence of smoking in people who live with HIV is higher than in HIV negative populations. And they also smoke a lot of cannabis. So 39% of Canadians living with HIV currently smoke cannabis. A lot of this is for recreational purposes. And what that impact has on the airways, it certainly is a contributor, but I would say that if you look at any epidemiologic study, the risk for having COPD, even after accounting for smoking, still remains high. So there's something in addition to smoking. It's not just a smoking effect. Now, smoking can have a synergistic effect on the HIV airway, at least in animals. So this is a monkey model in which they expose these monkeys to filtered air, cigarette smoke. Some of them may have been infected with a simian version of HIV, and then the combination of cigarette smoke and simian HIV. And you can see that there's this is the airway epithelium here. And you can see there's thickened mucus production, increased goblet cells. This is a stain for mucus. And you can see that there is some synergy between cigarette smoke and HIV. So that may be one part of the question. There's also some other unique aspects to the HIV lung, and one is oxidative stress. So medications like protease inhibitors, which is a powerful antiretroviral agent, can increase reactive oxygen species. And there are studies in the literature taking lung samples from patients with HIV and finding altered levels of glutathione and hydrogen peroxide. So that's one potential reason. And even if these patients are on chronic antiretroviral therapy and they have suppressed peripheral blood viral loads, there's still the persistence of latent reservoirs in the lung. You can't really get rid of them. And Dr. Kostiniak and Gill has measured this in lung samples. These are bronchoalveolar lavage samples here in the middle. And even if these patients are well controlled, you can still find virus in the lung. And what that does to the lung, I think we still have yet to figure out. The other interesting thing about this patient population is that they do get a lot of pulmonary infections. Even in this era of ART, they still remain at higher risk for things like community-acquired pneumonia. And while opportunistic infections, the ones that really attack the body when your immune system starts to get towards the AIDS, the AIDS range of CD4 T cells, things like pneumocystis, mycobacterium avium infection, that can affect the lung. And pneumocystis is an interesting one because they have looked at this again in a simian model, and those who were colonized with pneumocystis, so this is not necessarily the disease, but these are ones that they had been able to that they had exposed to pneumocystis, had more emphysema in their lungs. You can see this is the pathology of the lung. You see these dilated air spaces, kind of similar to my patient, in fact, compared to the monkeys that did not have any pneumocystis. So there may be a contribution from that. Now, in my lab, I've been interested in aging because if you look at this multimorbidity list in HIV, the thing about all these diseases is that they are diseases of aging, right? These are all things that affect elderly populations. And so one of the theories is that HIV actually represents this a phenomenon of accelerated aging. So this is what, aging is a complex process. It is it's the loss of homeostasis over time. And there's many factors that can go into aging. These are some of the pillars that we consider. So genomic instability, telomere attrition, epigenetic alterations, loss of proteostasis, mitochondrial dysfunction, cellular senescence, stem cell exhaustion. All these things can contribute to aging. And we've been interested in how to measure aging in patients living with HIV. And there's many different aging biomarkers out there. The easiest one that we all use in our daily lives is just chronologic age. But of course, we know that that's not a great measure of biologic aging. We always say, oh, he looks older than his age, he looks younger than his age, et cetera. So we have looked at things like telomere length, and these are the ends of the chromosomes that shorten as cell division progresses. And so the shorter the telomere length, that would be considered more biologically aged. And more recently, we've been looking at epigenetics and epigenetic clocks. So I'll kind of go through my journey through these biomarkers of aging. So when I first started off as a postdoc, we did look at telomere length in patients living with HIV and peripheral blood telomere length. We found that yes, HIV was associated with a shorter telomere length compared to non-HIV populations. Now it looked in this particular study, it looked as if the decline over time was actually similar between the populations, which suggested to us that perhaps there was some drastic hit that HIV patients took to shorten their telomere length. And then over time, as they get on antiretroviral therapy, perhaps that attrition stabilizes. And so we then partnered with one of the cohorts here in the downtown east side, just behind me here, of patients who were injection drug users. And we had samples of blood before they caught HIV, immediately after they caught HIV, and then several years after their HIV seroconversion. And in fact, right after they seroconvert seems to be the time when there is this most significant telomere drop. So suggesting that the immediate post-HIV period, something was happening in terms of that accelerated aging signal. So this was work I had done, earlier on, and I will, telomere length is not the greatest approximation of biological age. It only has a correlation with chronologic age of about 0.3. So we were looking for more sensitive measures of biological aging. And so more recently, we've been interested in DNA methylation as a way to quantify accelerated aging in HIV. So of course, this is the dynamic process of adding or removing a methyl group to a CPG site along the genome. And this removal or this addition of the methyl group can affect gene expression, either blocking it or activating it. And it's a way in which, because it is sensitive to things like environment, toxins, inhalations, exposures, it is a way in which the gene and environment can interact. And there are these certain changes that can occur, predictable changes that can occur in your methylation patterns over time when you move from being young to being elderly. And so there will be certain sites that may be demethylated or methylated as one ages. And overall, there is this global hypomethylation that occurs along the genome, particularly along transposable elements. So things like line 1 and ALU, which make up a large chunk of our genome, tend to over time get hypomethylated with age. So we've been interested in looking at the methylation status of these transposable elements as a marker for aging. The other thing is that because there are these predictable changes, you can create these clocks. And so this is our collaborator, Steve Horvath at UCLA, who's been really the brain behind these epigenetic clocks, where he has taken a weighted regression of methylation profiles and regressed it on chronologic age to create this clock that is actually a pan-tissue clock. You can see how good the correlation with age is from blood to liver, lung tissue. So it's been this pan-tissue clock that has been shown to not only identify people with advanced epigenetic aging, but also to help predict mortality and poor outcomes in a variety of different diseases and populations. Now, we've been working with Steve on some of this, and he created the original clock, which was a pan-tissue clock, and there are now different iterations. The Hannum clock is a blood clock. Steve has subsequently gone on to create other clocks that are trained on different aspects of aging. So the skin blood clock was trained on fibroblasts, works very well in patients, for example, with progeria syndromes. Telomere length was methylation patterns that were correlated with telomere length. Grim age was trained on smoking and inflammatory plasma proteins, and pheno-age on clinical manifestations of aging. So there are these various clocks out there that we have been using to see if we can identify accelerated aging in our population. So how we look at the output here is we basically regress the epigenetic age that we calculate from methylation profiles on chronologic age, and we look at the residuals. So a positive residual would suggest that you are older, you have an older epigenetic age compared to your chronologic age, whereas a negative residual would suggest the opposite. With telomere length, it's slightly different just because the shorter telomere length is more likely to reflect aging. So it's an inverse relationship. So I want to highlight, first of all, the work by Yolanda Yang. Yolanda was a master's student who had come from China to work in our center, graduated from her master's degree under Dr. Scott Kebbett, and then joined my lab as a bioinformatician, and has since moved on. She's now a PhD student under the supervision of Dr. Raymond Ng at UBC. But she was the first to take a look at epigenetic aging in early HIV. And so this was a collaboration that we had with a multi-center international cohort called the START study. The START study took patients who were very early on in their HIV infection, so still had preserved CD4 counts. They were asymptomatic, and they would randomize them to either starting HIV medications immediately or a late start, which means that they wouldn't start antiretroviral therapy until their CD4 counts fell below 350 or they had an AIDS defining illness. The way we got involved was there was a nested study within that trial looking at pulmonary function over time, and also a genomic study where they collected blood. So we had the intersection between blood and pulmonary outputs. And this was through a connection with our collaborator, Dr. Ken Kunisaki at the University of Minnesota. And so what Yolanda found was that, yes, HIV was associated with higher blood epigenetic age compared to a group of HIV negative controls. And what's more, there were a lot of methylation differences. So there were 56,000 CpG sites along the genome that were differentially methylated between those with HIV and those without HIV. And this, we're still talking early asymptomatic HIV patients here. So even early on in their infection, you can see that there were these profound methylation changes. And they were, if you run a pathway analysis, a lot of cancer signaling pathways and also some infection-related pathways as well. But the next step was really to define whether this was related to airflow obstruction. And this was the work of Ana Hernandez Cordero. She is a postdoctoral fellow in my lab. She is originally from Panama and finished her PhD in genetics in Scotland. And then we had the great honor of having her in our lab for the last few years. And she started to connect DNA methylation with airflow obstruction. And in fact, yes, if you go by, again, those ALU and LINE1 transposons, there was greater hypomethylation in those HIV patients who would meet criteria for having airflow obstruction based on, some of these are just lung function parameters up top here. So either criteria, there was this general hypomethylation. This is on the x-axis here, the beta difference between those with and without airflow obstruction. And so all of the blue here suggests hypomethylation compared to those without airflow obstruction. So generally there was this global hypomethylation with airflow obstruction in HIV. And again, she found there were 4,600 CpG sites along the genome differentially methylated between those with and without airflow obstruction. And interestingly, the pathways that come up, cellular senescence, mTOR signaling, P53, lung cancer, all things related to aging. So again, suggesting that HIV and lung disease is marked, at least in the blood, by an accelerated aging pattern. But of course, the question was, does this happen in the lung? All right, so we had all these blood samples, but we actually didn't know what was happening in the lung. And this is where our bronchoscopy program really played a central role. So St. Paul's Hospital is a very busy bronchoscopy center. This is a procedure that we do where we insert a camera down into the airways and we can collect brushings, biopsies, washings of the lungs. They're usually done under conscious sedation, and it's often an outpatient day procedure, but we do about 800 of these bronchoscopies every year. And when I was a postdoc, I started adding on or consenting patients for research bronchoscopies. And patients have been extraordinarily generous with their time and their bodies to contribute specimens for our research. And so we collect airway epithelial cells, bronchoalveolar lavage. We do oral washes. We collect blood from them as well. And so it's become a very rich biobank for us to use. And this was initially started just specifically to look at HIV lung disease. And so a few years ago, our master student, Stella Zhu, who came to us from China, we did take a look at telomere length in small airway epithelial cells from patients who had undergone bronchoscopy. And she was able to find that there was decreased telomere length in these airway epithelial cells in people living with HIV. But at the time, this was a few years ago, we still weren't powered yet. We didn't have enough of the sample size to really see whether the combination of HIV and COPD had an even faster accelerated aging pattern. And so this has been the work of Anna over the last couple of years. We just got this published last week in the American Journal of Respiratory and Critical Care Medicine. Finally had enough patients to look at airway aging and methylation disruptions in HIV-associated chronic obstructive pulmonary disease. And so it took us a long time to collect, a sample size of, 16 to 22 in each group. And so these are combinations of HIV and COPD, all of whom had gone to do a bronchoscopy. We collect airway epithelial brushings that we profiled for DNA methylation and RNA sequencing. And these are just details of our analysis pipeline. We ran epigenetic age, and for sure that HIV and COPD are both associated with advanced epigenetic aging. But really, it's that combination of if you have HIV and COPD, you had the fastest age acceleration of all of the groups. And when we integrated the methylation with the RNA-seq, we're looking at the corners here of the most significant both methylation and expressed interactions for people with both HIV and COPD. There is this distinct methylation profile. These are some of the genes that have come up as significant. There are almost 8,000 differentially methylated sites that are associated with the interaction of HIV and COPD. This suggests that there is this distinct molecular profile to this group. This is not just, it's not the same as COPD in the general population. This is it's a different form in their airway epithelium, a different kind of behavior. And if you look at the pathways that are enacted by these methylation sites, again, you see the same sorts of pathways coming out: cellular senescence, longevity regulation, mTOR signaling, PI3AKT, MAPK signaling, insulin signaling, all of these things having to do with age. And again, lung cancer comes up as well. So these are some of the top pathways that are associated with those differentially methylated sites. So again, it's kind of the same pattern. So which we found in the blood, we can now find in the lung. Recently, Anna has been looking at whether these epigenetic age biomarkers can predict poor outcomes in HIV. We just submitted this paper this week. And so we've been looking at this biomarker GRIM age, which again was trained on not just methylation and chronologic age, but also smoking and inflammatory proteins. And she was able to find that your lung function decline over time is related to GRIM age. So the more decline that you had in this corner, the higher your GRIM age acceleration was. But interestingly, patients, these were blood samples from the START trial. If you did not have any lung disease at the beginning of the trial, but over 6 years developed airflow obstruction, we could detect actually a higher GRIM age from the very beginning. So it may have some, it may be some clue that this patient may be at increased risk for developing lung disease down the line. So the question really now is, can you actually turn back this clock? And it's, it's an intriguing area, xenotherapeutics in HIV. If this is, normal aging here, and this is aging with HIV, can we actually turn back the clock with xenotherapeutics, geroprotectors. There are a number of compounds out there that have been, may play a role in reversing some of these aging changes. It's still a little early, but if you look back at our, the pathways that we came up with, some of these do have therapeutic agents already attached to them. So metformin for insulin signaling, rapamycin for mTOR signaling. Our colleague Steve Horvath did actually do a kind of a controlled trial by giving patients a combination of metformin and human growth hormone and another hormone, and actually found that you can, revert your epigenetic age, so that, you can decrease the epigenetic age. Now, what this means for clinical outcomes, I don't know yet, but it is kind of an interesting concept in whether we might be able to revert the aging process. And if there was a population where this would be really interesting to trial, it would be, patients living with HIV who have this accelerated aging phenomenon. So all very preliminary, but I think it's an interesting sort of future directions for this work. So where are the gaps? So, we don't know whether this is cause or consequence. A lot of our studies are still cross-sectional. I'd love to be able to do longitudinal bronchoscopies on people, but of course that demands a lot of an individual, and we still haven't quite got there yet. When we can detect this aging insult, my suspicion based on our work is that it's early on. But again, if that's the timing of intervention, I'm not sure yet. I think this still does need to be clarified. Is this an HIV specific aging process, or is this just accentuation or acceleration of normal aging? I think we still have to define that. And what I think is interesting is, what are the implications for the general population, right? If this in HIV is accelerated aging, are these things that, are these pathways still relevant to regular COPD? Are they just turned on faster in HIV? And if we're able to target these pathways that we found in HIV, could that mean that we can prevent the sort of disease that we see in the general population? So I think those are interesting questions that we still have to answer, and my lab is working towards that. Okay, the last part of my talk, I did want to just go a little bit into microbial dyspepsia. So we've been very interested in the microbiome, mainly because these are patients who do get exposed a lot to pulmonary infections, and as a result, they do get exposed a lot to antibiotics as well. And so we've been very interested in sort of profiling the lung microbiome in HIV. And again, Stella had done this as part of her master's degree, and she did discover in the small airway epithelium reduced bacterial diversity here. This is the Shannon Diversity Index, and there are these phylum changes when you go from HIV negative to HIV positive patients. But of course, it was early, and we still had not enough samples to really look at COPD as it relates to HIV. So this has been the work of our master's student, Marsha Jude. She just graduated at the end of last year. She came to us from India and has since graduated to become a bioinformatician in one of the labs here at the Children's Hospital. And she had been working as part of her master's thesis on the microbiome of HIV-associated COPD, integrating it with other omics outputs. And so she found that in comparison to all three other permutations of COPD and HIV, those with both COPD and HIV have the lowest microbial diversity. And diversity is often a hallmark of health. So the lower this was, more of a sign of disease here. And what's more, the bacterial composition was quite distinct. And so if you plot out the PCOA on this side of the graph, the purple here, your people with HIV and COPD seem to separate significantly from a statistical standpoint compared to the other groups. And what was different here, I just want to focus here on Prevotella up here, which is one of the genus that came out as most significantly different between these groups. You can see this gradual decrease in the COPD HIV group of Prevotella. And where that becomes interesting is when you can start to integrate these outputs with now the methylome and the transcriptome. And so she had all three omics platforms to work with, and could do an integration analysis using a program called Diablo and find these connections. So what does it mean for Prevotella to be decreased in this population? What does this actually correlate with in terms of methylation or transcriptome? And she was able to find that, you know, this kept popping up. Prevotella, it had this relationship with this gene fuzz, which is a ciliary gene, cilia being very important in the small airway epithelium to move toxins and bacteria out of the lung. And fuzz kept popping up in the transcriptome as well. And so you can start to create these networks to understand what the impact of microbial dysbiosis is in this population. And these were the top correlations that she found that were associated with that interaction between HIV and COPD. So Prevotella, fuzz in both methylation and transcriptomic output. So this is, we're getting ready to submit this paper. I think it's a really unique way to look at what the impact of microbial dysbiosis is in HIV, in the lung. So where are we going from here? I'm just gonna wrap up in the next few minutes. I wanna highlight some of the work done by three people. So Rachel Eddy is a postdoctoral fellow at our center who came to us from Western University after finishing her PhD there. She is a Michael Smith and NSERC trainee and is heading up an imaging core here at HLI. I also want to highlight Stephen Milne, who is a respirologist from Sydney, Australia, who's doing his postdoctoral fellow here, and Firuza Gara Yali, who is a PhD candidate here. We're very excited because Rachel has been spearheading this, our new MRI imaging core at HLI. So this was installed last summer. We have a dedicated MRI machine here at our center just for research purposes. And why we're really excited about this is because when patients breathe in a xenon gas mixture and we run them through the MRI, we are able to see ventilation defects. So this is actually my patient with asthma, and everything in cyan here is normal. Everything in black is abnormal. And you can see here with greater resolution than CT, in fact, all the areas where his ventilation may be impacted. And what Rachel is able to do, she's able to take the CT from this patient and merge it with the MRI so that we can get not just some of the structural changes on the CT, such as emphysema, airway loss. We can start to tie that in with where there may be defects in ventilation. And we're further then able to tie that in with my bronchoscopy platform. So I'll just give you an example of what we've been doing lately. This is a 50-year-old patient with HIV on antiretroviral biotherapy, but she does have COPD. She underwent a CT scan and an MRI. And Rachel was able to merge those two images together to tell me where on the CT in yellow she had areas of emphysema and where on the MRI she had areas of ventilation defect. And she's able to quantify that for me. She tells me, okay, in the right upper lobe, she has very bad emphysema and ventilation, but in the left lower lobe, she's pretty good. So I then take my bronchoscope and I collect samples from diseases of, diseased areas and non-diseased areas. We're able to do single cell sequencing, comparing those two regions, and I can start to, this is still very preliminary, but I can start to see on the same genetic background what might be driving disease up in one area of the lung versus, a less diseased area. So we're very excited about this because it's really trying to integrate imaging and our sequencing platforms now. The other thing that I'm very excited about is optical coherence tomography. So this, we just got this into our center. This is a catheter that can go through my bronchoscope, and it uses near infrared wavelength to actually reflect back and give me at least 2 to 3 millimeters of image around the airway where I'm brushing. So the way this works, this is a picture of our bronchoscopy suite. Conventional bronchoscopy, this is the picture of the airway. This is a patient with HIV and COPD that we were doing a bronchoscopy on a few weeks ago. And when I take my bronchoscope out and push a brush out, I am blind essentially. I can only see this part of it. When I push my brush out to the area where I take the brushings, I guess at what that airway looks like. Rachel can tell me that that area is diseased or not diseased, but I actually don't know what the configuration of that airway is when I brush. But with the addition of the optical coherence tomography, I can get an in vivo image now where I can actually take my brush and see what the airway wall looks like. And so it can give me a reading of the airway wall, exactly where I brush. I can see how thick it is, what the wall area is compared to a normal looking airway. We can start to look at pathology. It's kind of, yeah, it's like a in vivo histology that we can do in the bronchoscopy suite. So like, you know, here's my bronchoscope here, and I can get a good real-time image of the airway that I'm collecting. And so we're very excited about it. We did our first case on April 7th. I'll just say, you know, this machinery took 4 years, 2 grants to get to our center. We had all of our stars aligned finally on April 7th. I found a patient, you know, again, with HIV and COPD, who not only was willing to let us put him to sleep and put a bronchoscope down his airway, but also was totally cool with us saying, hey, we're going to use this catheter for the first time in our center. Are you okay with that? And he was totally game for it. Yeah, we got all of our stars aligned. I you know, flew in a team from the company to help us out. And of course, you know, a few days right before this happened, I tested positive for COVID, and they had to zoom me into the room. And so I was rather disappointed not to actually, these are pictures from my from the Zoom screen that I had. So yeah, sometimes life just gets in the way of research, but we are very excited about this because I think this is kind of a new era of airway phenotype. So other things that we are doing in our platform. So the nice thing about having a bronchoscopy-based platform is that we can, you know, look at, you know, it's very translatable to other diseases. And so when COVID hit, we had this biobank of airway epithelial samples that we had already profiled for RNA-seq, and everybody was talking about ACE2, and we just, hey, you know, we just decided to do a quick analysis of our database, and we published this, I think, in April of 2020, like fairly early on in the pandemic, and looking at ACE2 expression in COPD patients, and found it was elevated, particularly in current smokers. This has since become my highest cited paper, just with the nature of COVID, but it's nice to kind of repurpose a lot of our biobank for, you know, very urgent sort of public health matters. The other thing that we're doing right now, we just launched this. This is our Connect study. I'm the PI of a longitudinal study on cannabis smoking, and we are again doing imaging, MRI, CT, bronchoscopy, single cell sequencing on people who smoke cannabis, comparing them to tobacco users and patients who don't use any inhaled substances. So we're very excited about this. This is our study website if any of you are interested in finding out more. I just wanted to thank your attention for today, and to the organizers for having me here. I really want to thank all of my graduate students and all of our collaborators. It really, like I said, it takes a village, and, you know, I have interventional bronchoscopists, HIV specialists, HIV researchers, study coordinators, radiologists, you know, a lot of people to thank. But most importantly, I do want to thank our patient participants, right? It takes a special person to say that he is willing to undergo a procedure, an invasive procedure, for no other reason than to help, you know, his fellow mankind. So I do want to thank all of them for all that they have contributed to our research. So I'm happy to take any questions, yeah, thank you so much.
Well, thank you so much, Janice. So first of all, I'm really sorry for being late.
That's okay. It's nice to see you, Emmanuel.
For some reason, I had this in my calendar as at 12:30, and I think it rather ends at 12:50. And, and so, yeah, there you go. Oh man, it's amazing, the nostalgia, just seeing all the data and all this, the story. For those that do not know, obviously, Janice is from my former center, and a senior colleague there. And obviously, there's the bronchial brushes that she takes, the epithelial cells that she takes is an amazing resource actually for some of the work that I've done in the past too. So this is amazing. I would open, so I'll take over and open the floor for Q&A. If anybody has any question, they can unmute and ask, or they can put them in a Q&A session and I'll read them out. So Robin, the floor is yours.
Sure. I thank you. That was wonderful. It's so, I agree that imaging is spectacular. It's a really, you've got a really cool setup.
Thank you.
I have one question that's sort of related to the research and one's a little bit of an aside. Do you break down your database in terms of additional factors? Like you have HIV and COPD, like health factors, but like demographics or socioeconomics or any of those other things?
Very good question. I would love to. As you can see from our numbers, and the amount of effort it takes to get to build a bronchoscopy corridor, we're not huge, right? We're not talking like hundreds of patients. I think it's hard to do in terms of the airway epithelial samples. I think blood, if I had the START cohort is much larger. We have peripheral blood samples. They have much better phenotyping of social demographics. We have not yet. It would be very interesting to see because epigenetic aging has been shown to be related to a lot of things like poverty and a lot of different demographic factors that I have not looked at yet. I think that would be awesome. I would probably need some help as it's not my area of expertise, but I think that would be very cool to do. I think in our cannabis study, we have taken the effort to really try to capture that information, things like economics, employment status, gender identities as well. I think we should be able to do that in our cannabis study on a larger scale. Definitely something I'm looking forward to seeing the data and seeing how it works. It'll be several years before I'll figure that out, but we've at least tried to ask patients those kinds of questions and participants those kinds of questions so we can hopefully do that in the future.
Awesome. My second question is a little bit of an aside because it triggers, you were talking with so much gratitude about your patients who have offered up samples. In my teaching, we talk a little bit about ethics and what good ethics might look like and what breaches of ethics might look like. And so I was wondering if you could just speak to, I'm running on the assumption this is a good ethics, of course. So can you speak to sort of what the process is for sort of getting the permission to get those samples and what the patients go through?
Yes, absolutely. So it's hugely informed consent. Right? You have to know what you're signing up for. This is not a blood draw where, it's pretty routine. This is an invasive procedure that does carry risks, especially in patients with COPD. We're also not talking about healthy controls here. We're talking about patients with lung disease, right? And so with every patient, I go through all of the risks and benefits, right? All of the potential complications that could come out of this procedure and what the benefit to them might be. And the benefit is not obvious, right? The benefit is not tomorrow you're going to find the answer to the disease that you have. The benefit is theoretical, that I'm hoping that one day I will figure out the disease that you have. It may not be today, may not be even in your lifetime, like these are incremental steps on the way. Now, I work with a patient population. I think that tribute to the HIV community who has been part of research from the very beginning, right? Like, the reason why ART has been, it took patient advocacy and, and patient engagement to really get those kinds of discoveries. And so they are wonderfully engaged with research and more than generous, and understand, I think, the importance of patient contributions to research. So to be honest, I've never had resistance to this. In fact, if anything, I've had people go, hey, sign me up. Whatever you can do to figure this out, sign me up. And of course, it's completely option. A patient is never coerced into this. It's never part of their if they're not comfortable, they don't participate, right? It's all about informing them of the risks and the benefits. And I will say that patients have been wonderfully generous, and they buy into the importance of research for sure.
Awesome. How specific, like you said, for example, that you sort of had the opportunity at the beginning of the COVID to sort of use some of those samples to look at ACE2. So how specific, like, do they know exactly what study, or do they know that it's just forming part of this database? Like how specific do you have to be in that consent?
Yeah. So we do say that we can, there is a clause in the consent that it may be answering additional questions, that we may be using samples with other researchers who may not be directly related. So we are able to kind of repurpose this for future use. We have kind of a future directions, future use clause in the consent to help with that. Yeah.
Awesome. Okay. Thank you.
Yeah. Oh yeah, Shelley has a question.
Hi, Dr. Leung. Thank you so much for your fantastic talk. I really enjoy it. My current research is about neurodegenerative disease. So I want to ask a question relevant to it. As you mentioned, COPD is a common respiratory disease among elderly. Also, COPD is a type of systemic inflammatory disease. Inflammation can contribute to a lot of disease, such as neurodegenerative. So do you think patients with COPD have a higher risk to develop neurodegenerative disease when they reach a higher age?
Patients with HIV, sorry, I heard part of it. So are patients with HIV more likely to develop neurodegenerative disease? Is that the question?
Sorry, patients with COPD.
Oh, COPD.
Yeah.
So, I mean, it's a little, because patients with COPD are elderly, so age is just the common risk factor here. There's data to suggest that there are some functional brain changes in COPD as well. So I don't know whether that is heightened in HIV. To be honest, I don't think I've ever seen a paper. I myself have not looked at whether there is a synergistic interaction between HIV, COPD, and neurodegenerative disease. I can't recall off the top of my head, any study that has looked at that in particular. I think it makes sense in the sense that if there is a common inflammatory pathway, it's systemic and it may relate to neural tissue as well as the lung. That makes sense to me, but I don't think anybody's actually looked at it. I certainly have not looked at it yet.
Okay, thank you. My second question is that there is a growing amount of research on COVID-19 in the recent years, and you also published some articles relevant to it. So I wonder, is COPD a risk factor for COVID-19?
Good question. Yeah, okay. So one of the striking things, like very early on, if you look at China, Italy, the US, it was, hey, how come COPD patients are not really showing up in the comorbidities? It's not like obesity, hypertension, diabetes. All of those things were very, very highly associated with COVID-19, but not COPD. And so nobody really knew why that. Maybe it was underreporting. Maybe nobody knew who actually, we know that COPD is just underreported in the general population. People don't get pulmonary function tests like they should. Was it that they know that they have lung disease and they stayed home and they wore masks and they did they did what public health ordinances told them. Maybe it was that. So I'm not sure that they are more susceptible to actually contracting it. But if you do contract it, if you have COP, you are more likely to end up in an ICU, have a poor outcome, die. So there's a difference between susceptibility and poor outcome here. It's not clear yet to me whether there is some degree of protection from inhaled corticosteroids that they may be on as well. That's one of the possibilities. I would say that it's not a good thing when they get it. I still recommend all my patients, with pretty severe lung disease, continue with masking and obviously get vaccinated. But I think the susceptibility piece, I'm not sure they're more susceptible, but to getting it, but certainly having a poor outcome for sure.
So like they either have very good prophylaxis,
maybe,
or they have a very serious symptom, so there's no on recall.
Yeah.
Nice, I understand. Thank you so much.
Yeah, that goes directly to, I'm smiling because I know, yes, I'm hearing from Janice, and that means what I've been telling people is okay or is correct. But people have been asking me about this whole COPD conundrum. And I'm like, I'm not a clinician scientist, so I might not be the very best person to answer this question. So man, all the imaging facilities that you have now.
Yeah.
Amazing.
Yeah.
I'll probably be coming back to see you guys.
Well, yeah, no. If you ever want to collaborate or have any ideas, we're happy to keep the conversation going for sure. Yeah. Absolutely. Anything we can do to help your program out there. Yeah.
Yeah, and for me, the connection between the MRI and the CT, immediately I saw that my mind obviously went to micro CT, but then to think about, okay, micro CT is not on living tissue, but is there any kind of thoughts to go in that direction where you actually are kind of really having that big resolution with micro CT in addition?
Okay, well, this goes back to Robin's question about ethics. Okay, so the bigger the tissue sample that I get, the happier Emmanuel is as a reviewer. The happier Emmanuel is, but we increase the risk to the patient. Yeah. So we do not get big biopsies from patients. We could potentially get smaller sort of endobronchial biopsies, but certainly something like a transbronchial biopsy, I think significantly increases the risk to the patient. And so we have not done that. And, those would be things that potentially you could run through a micro CT. But right now we mostly have just, cells that are in suspension as opposed to like a structure that you can, now, one thing that I will say that may change things is that the better we can control HIV, the longer that these patients can live, the more likely I think in the future that they can undergo things like a lung transplant. Now, it used to be that patients with HIV could never undergo a lung transplant. It was an absolute contraindication because people would say, oh, you immunosuppress them, they'll lose the lung. But that may change. And there are definitely centers that have successfully transplanted well-controlled HIV patients. And so if that becomes more of a standard in the future, we may be able to get lung explants and really start to do, some of the work that your lab and, others have done with micro CT and understanding the structure of the lung. We just haven't been able to do that yet because that hasn't been standard of care.
Yeah, that's really great. And I mean, as we wait for that, as you're talking and getting all these tissue samples, what comes to mind obviously is building models that we can then use to, we can use for these imaging studies. So obviously I have a ton of questions, but I'm just going to ask two. Oh, you have a, sorry, somebody else has a question somewhere?
You go first. I have a question, but you go first.
Okay.
But you go first.
I was just going to ask two of those, the cause and consequence, hold the hypothesis or the, when you're talking about whether aging is a cause or effect when it comes to HIV and COPD and the differences between HIV COPD patients and just normal COPD patients. And the fact that obviously the data that you have is cross-sectional and not necessarily mechanistic. And so we are not able to really determine, you know, are you able to speak to that a bit in terms of how we'll be able to answer that question?
Yeah, okay. So ideally, in an ideal world, I would love to bronch a patient. What I would need to do is I'd need to bronch a patient before they capture HIV, immediately after they get HIV, and then when they get on antiretroviral therapy. Again, back to Robin's concern about ethics, there's a lot of problem there, right? Okay, one, it's very difficult to capture patients nobody knows they're going to get HIV, like, you know, pre-HIV versus post-HIV is very difficult to do. And it can only really be done in high-risk cohorts, like we did with the injection drug users, right? Delaying someone's ART so that I could do a bronchoscopy and study them is unethical, right? We know that now from the START trial. So it's tricky, right? I mean, ideally that would help to answer my question. Now, I think longitudinal bronchoscopies would still be good to, you know, what happens with smoking over time, or, you know, different sorts of exposures. I think you kind of have to turn to the lab again and start doing in vitro models of exposing cells to HIV, exposing cells to cigarette smoke, and kind of starting from there before you go to your primary, you know, human samples. Those are some things that we have been working on, trying to do a co-culture model of airway epithelial cells and HIV infected macrophages. It's tough, it's hard. We've been struggling on that. You know, I think, you know, I've definitely had, you know, students kind of spend a lot of time, you know, going nowhere, doing stuff like that. But I mean, I think those are some of the surrogates that you can use. In the absence of having the human samples.
And is it are comparative studies between obviously COPD patients without HIV patients who are COPD vis-a-vis the time of infection and obviously the duration of the COPD, do those hold any merit in these kinds of conversations? So comparing, you know, in between these different populations that are obviously just having an intra-patient comparative study.
Intra-patient. Well, I think the START study is probably the best. Well, the problem with the START study, unfortunately, they did not capture longitudinal blood. Because that's the other thing, right? Like, they followed them for many years, and it would be ideal, like the ideal population to look at pre-HIV, you know, pre-antiretroviral therapy, post-antiretroviral therapy, and changes in the blood with that. But unfortunately, I think they only collected, they may have collected plasma, but I don't think they collected like DNA. So it's difficult for me to answer the methylation question, but something longitudinal like that, I think would be helpful. Yeah.
Okay. And the last thing is, you know, obviously the study on the microbiome, you know, amazing stuff. And I'm actually getting interested in that right now, trying to understand the gut-lung axis. I would love to understand how, you know, obviously microbial populations in the gut affect the lung, and vice versa. First of all, are you able to speak to that in terms of like
a little bit? Okay,
I'll add one more to it. You talk about is it Provitella? What does it do in the lungs?
It's an oral. Well, okay, so it's mostly an oral commensal.
Okay.
Yeah, so maybe there is some sort of maybe there. I mean, there's obviously crosstalk between your oral cavity and the lung as well, but it's mostly an oral commensal that you know has been implicated in COPD before as well. So it's kind of an interesting microbe in that sense. But again, its role in the we're not really sure yet, and I think that's why we need to sort of do these integrative studies to really look at the impact on the lung. So your other question,
gut-lung axis.
Gut-lung, right, gut-lung, right. So it's very interesting because HIV gut mucosal leakage is is kind of a hallmark of the initial HIV infection, and so that's probably in some part related. Now we had these. So unfortunately, we did not collect like stool samples from our patients. It would be very interesting to look at that connection. We did have autopsy specimens from an HIV cohort in the United States that we ran microbiome. We had lung and we had gut samples as well. And some of them were paired. We haven't published this yet, but we did look. There wasn't we couldn't find too much relationship between the two of them, but it was a very small sample size. So I don't want to say, I mean, I would love if we had lung and stool samples. I think that would be really, really good. I wish we had collected that, you know, in retrospect now, because I think it is a very interesting question, because I suspect that there is crosstalk, and that there, you know, microbial dysbiosis in the gut is likely probably going to be related to the lung. We couldn't show that in our tissues, but I will say that, you know, autopsy tissues are not really preserved for microbiome, or there's probably a lot of contamination, and we know that that's the contamination in the lung, contamination in the gut they can handle, because there's so many, the diversity and the species richness is so much. In the lung, it can totally mess things up because it's a low biomass sort of organ. So yeah.
Interesting. Lots to think about. Robin.
Yeah.
Wow, I'm just actually quite enjoying while listening to the two of you like riff off each other on this stuff. I actually want to completely change gears and ask you a little bit about your journey. Because being a clinician scientist is a long and arduous journey, as far as I know. So can you speak to a little bit about how that was, what challenges? Things like that?
Okay, yeah, absolutely. Yeah. So when I finished my clinical fellowship, I came specifically here to Vancouver to HLI to do a research postdoctoral fellowship. And that was a huge learning curve for me. Like, you're talking to somebody, I do not have a PhD. I do not have a master's degree. I only knew my little world, my world in the hospital. And so all of a sudden learning how to design an experiment, how to design a study cohort, how to write a grant, you know, all these things. Like, I literally started from scratch, and it was a lot of trial and error. I mean, if you saw my first grant application and the sort of truck-sized holes that like the reviewers, you know, it was just a learning process. And, you know, the good, like Emmanuel knows, like there were great people around our center to mentor. It's really about mentorship from, you know, for me, it was Donson and Paul Mann who really mentored me and were, you know, kind of showed me the ropes in that sense. And you need an infrastructure, you need people to support you, you need people who are willing to take that chance on you, even though you're asking like the most elementary of questions. But it was, and I'll say it's still a learning curve for me, especially, you know, mentoring graduate students when I didn't actually go through, you know, a formal graduate school experience. So a lot of that has been trial and error, and you know, I definitely piggyback off of people, and if I don't know things, I can find somebody here at UBC who does know it and can help me out, and that has been critical, I think, to moving forward.
Yeah, I actually think some of the same things about being at UBC is a very good place to be for things like that. There's so many people around to help. Yeah,
absolutely. Yeah. And a lot of collaborators, and, you know, a lot of help along the way, for sure. And I do think it is nice that my clinical interest is the same as my, you know, I can talk to patients and have them inform what I do. I have access to them, you know, for their ideas, and they're always interested in the research as well. So there's a lot of, you know, I think it does kind of play off on each other to kind of work in both worlds. But yeah, it's almost like you can work in both worlds, but you're definitely not an expert in either of them. You know, you kind of straddling that. You kind of have to accept that you're not going to be the expert in either world.
Just
something. Go ahead.
I was just going to chip in there as to like what made you decide to do the postdoc, like the fellowship, after you did your clinical studies.
Yes. So you know, to be honest, I've always been interested in infectious diseases. And when I was trying to decide what kind of specialty I wanted to do, it was a decision between infectious disease and respirology. And so this was kind of a way where I could kind of do both and kind of merge those interests. And it was the right center to do it. Like I said, like there's a huge HIV center here that is world renowned, and also a fabulous world renowned lung research center. So it was like the kind of the perfect place to be to study that. And so I think there was a patient population here, there was the research infrastructure here, there were the two centers that worked and have worked very well together in the past. You know, so I don't know if you guys, Julio Montana, who is, you know, the bigwig here, is actually a respirologist. You know, that back in the day did work with Jim Hogg, and you know, so there has been a long-standing collaboration between these two centers, and so it's the perfect place really for me to be. Yeah.
One of our other talks was about sort of conservation and sort of the connections between Indigenous communities and how they can inform conservation strategies. So something you just said sort of made me think of that, about how your patients sometimes inform your research and vice versa. Like how much does that, how involved are they in sort of the research questions that you ask? How connected is that relationship?
Well, so you remember that patient CT scan that I showed
you,
right? Yeah,
yes.
That particular patient, when you look at that, you're like, how is this possible? How is this possible in a 45-year-old? And so that, you know, when you see something like that every week in your clinic, or, you know, multiple times in your clinic, then you start to say, okay, well, this is not normal. Something's going on here. What might it be? And, you know, he's actually a patient that we did collect. He allowed us to collect some samples from his lungs to really identify that. So they do. I also work with another patient who has HIV and has had HIV and COPD for a very long time, but is on my CIHR grant as a patient representative and helping with advertisements and how to reach out to patients and to do knowledge translation activities. So, you know, she reads over my grant applications, and she, so they are part of this process. Awesome. And I like that. I like that sort of back and forth. And I think they find it fulfilling too, especially since it directly relates to them and their condition, and you know, anything they can do to help feel better, they're supportive of.
Awesome. That's wonderful. Thank you so much.
Yeah.
And I think one thing that I also wanted to draw people's attention to was the time you spent in talking about the diverse people that you work with from the center. And when I first came to the center, I think at the time I was the only Black person, and then over the next 2 or 3 years, we seem to have like an explosion and a number of people from diverse backgrounds coming, to the point that by the time I was leaving, we're starting to have Black History Month.
My lab, I make a point, you know, we are very diverse. You know, you can see that I have, you know, my students come from all different walks of life, from areas around the globe. And I think that's important. I do try to make a point of keeping my lab diverse when I choose students, and try to have a range of perspectives. And I think it works well. I agree. The center is very diverse, and we welcome people from all over the globe.
Yeah. So my question sort of follows up on this. You've talked a lot about the rich opportunity set and the people and resources around you that have enabled this work. What barriers?
For me, barriers to my own success is that kind of, it's hard juggling a clinical practice with a lab. Clinical practice does not get lighter, it just grows. This is especially true in the pandemic, because we're now starting to see the backlog of patients. And even though one says protected time, in big quotes. So for me, it's hard to, and patient, you cannot wait, right? I cannot just say, okay, I'll deal with this patient in a couple of weeks when my grant application is done. I can't do that. So time management has been quite difficult. Again, like I said, I think it has been a learning process in how to, for someone who has not come from a graduate school experience, it has been a learning process of how to mentor graduate students. I'm still not sure if I'm doing it correctly, or ideally. Because it's a whole different experience for me. It's not something that I ever experienced. And it's different from mentoring residents in the clinical world. It's a completely different set of skills. And that has been a barrier to me. You know, kind of in retrospect, should I have gone to graduate school? There are days when I definitely think, yeah, I probably should have developed skills that would help me today, knowing, you know, we talk about biological age, like, you know, I have, you know, adding more years to my training, it's a toss up. So that has been a struggle to me, and I'm still learning that process. I think those have been some of the challenges that I have faced. I would love more time to just, you know, do my research, but I think just kind of balancing that with being a doctor in this age has been, and, you know, and in the past few years, you know, we've been called upon to do additional service because of the pandemic and everything. So it's been a little bit hard to focus, I think. It's a new age. Even my research platform is something that is very vulnerable to a pandemic. I stopped for a year, essentially, because I cannot bring a patient to the hospital. I cannot do an invasive procedure on a patient in a COVID, you know, in a pandemic. So that has been an additional challenge over the last couple of years, is how do I maneuver this, and how do I maneuver graduate students where, you know, I promised them this would be a great project, and all of a sudden for a year I stopped collecting samples from patients that they needed, you know. Yeah.
Awesome. Any other questions from anybody? Okay. Yes. Oh yeah, Shirley.
Yeah, I'm not asking questions, just like speaking about diversity, I also want to ask something before the close of our very last seminar. Dr. Louis Pasteur once said that science knows no country because knowledge belongs to humanity, and it is the thought which illuminates the world. And I want to quote and change this sentence. Biology knows no ethnicities or race, nationalities or origin. Biology has flourished not only because of biodiversity, not only because of diverse research from different areas, but also diversity of our human beings. Thank you.
That's great. I will probably have to steal that. So
we just have to put Shirley's name underneath.
Oh yes, of
she
course,
gets,
yes.
You know,
Science, yeah.
Forever from now credit. Louis Pasteur and Shirley Yang.
Thank you.
So, in absence of more questions, thank you. I'd like to thank you, Janice, so much for joining us.
Yeah, no, thank you for having me.
Sorry for being late again. Amazing stuff that we all, you know, before learned about, from science to your journey, incredible, and even talking about the diversity of in the center, great stuff. And also, you know, I'd like to appreciate and thank our able-bodied panelists and organizers who really have been able to put this into motion for this term. Actually, first time we've done this, and I think it's been a resounding success. And I look forward to attending more in the future. And yeah, and thanks to our audience and those who you know, tuned into. We were a relatively small audience for today. Totally understandable. We've done with our last exam tomorrow, so that's totally understandable. Thank you so much, Janice, and thank you.
Thank you, guys. Thank you, Emmanuel. Thank you, Robin. And thank you, everyone.